welcome everyone. This morning I have the honor and pleasure to introduce Dr. Laurence Beck. He's an associate professor at the Boston University in Boston. He's an excellent researcher and very well known for all his investigation in membrane nephropathy and all the new insights he has provided with this finding of the PLA2r. So good morning, Dr. Beck. I'm very, very honored to have you here, and thank you very much for accepting to contribute to our course.
Thank you, Carmen. It's a pleasure to be interviewed by you. Please go ahead.
Yes. So, okay. So membranous nephropathy is a very, very important disease. We have many cases. In Latin America, we don't have as much as you have in Caucasian population, but also we have a lot of cases. And I bet we don't have a specific study in Latin America to see we have the same findings that you have in other populations, but probably we have. So, but anyway, the information you have provided for us is very important for the treatment of these patients. So I brought a series of questions for you. So, and my first one is that I'm sure is not in the books. So what you are going to give today is different, not in the articles, is how you got the idea to identify the antigen responsible for the primary membranous nephropathy.
Mm-hmm. I wish I could take the credit for this, coming up with the idea, but it really dates back, and I can credit my mentor, David Salant, for this, because the story goes way back to the rat model of Hayman nephritis, which is a animal model of membranous nephropathy that was developed by Walter Hayman in 1959. And basically all these models were developed at a time where it was presumed that the antibody complexes that ended up in the kidney were due to circulating complexes, a serum sickness model. So it wasn't until the late 1970s at Boston University that David Salant and Bill Kauser and other coworkers basically disproved that hypothesis for membranous nephropathy by showing that antibodies that were injected into the rat in an isolated system where only the antibodies could circulate once, they basically showed that the antibodies were targeting an antigen in situ in the rat glomerular basement membrane, or maybe even on the podocyte membrane. So this was 1978, and it changed the focus in membranous nephropathy to looking for, instead of circulating complexes, looking for a protein that was on the podocyte. It wasn't until the mid-1980s that Dancho Kariashki and Marilyn Farquhar identified the protein in the rat. Initially, they called it GP330, but then discovered it was the big protein called megalin. And in the rat, megalin is on the podocyte, where it serves as the target antigen. But people quickly looked in humans and found out that megalin wasn't the target. So the search continued since the mid-1980s. And really the only next step that showed us that maybe there is a human antigen was Pierre Ronco's finding in Hanna Debiek's in 2002, that in a very rare case of a baby being born with membranous nephropathy, they realized that there were antibodies to the protein neutral endopeptidase. But this is a very rare finding and didn't explain adult disease. So that's basically where the idea came from, sort of searching for the human antigen. And when I came to the lab in 2003, it still hadn't been found, so that was one of my projects.
Wow. So yes, it's very exciting, all what has been done before by David Salant. Even when I was in Boston, I was working in membranous nephropathy, but we were working in the mechanism, and I was using serum albumin injected into rats, so just to identify, the charge of the molecule, and not exactly because we was an induced membranous, and this was very exciting. So, wow, it's very exciting. How do And finally, how did you identify that the PLA2R was the target protein? Because, I mean, I bet you may find that many other proteins there, right?
Yeah, so it's a long story, but basically, we and other laboratories were doing what you might expect, taking patients' blood, so serum from patients with membranous nephropathy, and asking, is there reactivity to any glomerular proteins? So how did we do this? We obviously collected blood from patients and from controls. And then for the glomerular proteins, we got kidneys, human kidneys that couldn't be used for transplantation. And we filtered out by mechanical sieving the glomeruli. And then we dissolved the glomeruli in detergent. So we made extracts of all the human glomerular proteins. And we ran them by gel electrophoresis, and we transferred to membranes, and we Western blotted with the patient's serum to ask whether there were circulating antibodies that might detect a human protein. Now, this technique wasn't, basically all other laboratories in France, in Manchester, England, that were looking for this protein were doing the same things. And initially, no one was finding any specific bands on the Western blot. So what was it that made us successful? One thing was, so it turns out that when you use the standard techniques for Western blotting and gel electrophoresis, you boil the proteins and you add chemicals that disrupt the disulfide bonds in the protein. It makes it easier to run on a gel. It turns out that when you do that to the target antigen in membranous nephropathy, you lose reactivity with the human antibodies. So one of the things we did was we ran the gels and we left those disulfide bonds intact. We just left out the chemical that disrupts them. And that allowed us to see a band. And the other thing is, we obviously have lots of antibodies in our body. Many of them are IgG1 subclass, and the ones in membranous nephropathy tend to be IgG4, which is the rarest of the IgG subclasses. So around that time, there were antibody reagents becoming available, so we could specifically detect the IgG4 antibodies in the patient's serum. And this gave us a very good signal-to-noise ratio on our Western blot. So basically, we either saw a signal or we didn't see a signal. So in about 50% at that time of the patients with membranous, we saw a band. And in all the controls and other glomerular diseases, we didn't see anything. So we had the band. That was the easier part. And then the question is, there are lots of thousands of glomerular proteins. How do we figure out which one it is? So we tried a number of biochemical purification techniques with columns, and ended up cutting out from the gel the smallest piece that we thought contained our membranous nephropathy antigen. And we sent it off to our colleagues in Louisville, Kentucky, at the University of Louisville, to do mass spec analysis. And basically, they just identified every single protein tryptic peptide that was in that little piece of gel we sent them, and then gave us a big list of all the proteins. And I'll certainly admit that PLA2R wasn't the first protein that we said, aha, that's it. Because there were a lot of podocyte proteins such as nephrin or alpha-3 integrin or podocalyxin and some other interesting ones that we went after first. And basically, we bought commercial antibodies, or we asked other investigators for reagents, or we got the recombinant protein. And pretty soon we recognized that all these interesting proteins didn't quite match up. So the band for PLA2R was a little bit bigger than all these other ones. So we still had work to do. And I won't go through the whole story, but there was sort of a chance finding. We were looking to see if the antigen could be cleaved from the podocyte. So we allowed the glomeruli to incubate at 37 degrees and asked if anything was shed into the surrounding medium. And it turns out it wasn't cleaved, but for some reason, the antigen came off into the supernatant. And when we reran that on the gel, it turns out that we had very well purified what turned out to be PLA2R. And when we repeated the gel electrophoresis, we repeated the mass spec, the number one protein that was on our list was this thing called the M-type phospholipase A2 receptor. And we quickly looked at the literature and said, what is this protein? We found out that it had been cloned by Gérard Lambeau in Nice, France. So we quickly emailed him and asked, he was a biochemist, I'm a nephrologist, we're interested in this funny disease, would you be willing to lend us your reagents? So he very generously sent us the antibodies he had generated to the protein, the DNA, plasmids, the recombinant protein. And once we got that package, we quickly realized that our patients with membranous nephropathy had antibodies that detected his protein. And that led to our joint paper describing PLA2R, or the phospholipase A2 receptor, as the target antigen in idiopathic membranous nephropathy.
Well, all this journey is very exciting. So my next question was about if there were other candidates, but you already explained there were others, but PLA2R was the one that reached the point about the band that you found in the gel. So it's very exciting. So, and my next question is, okay, we know now that PLA2R is there. What is is the PLA2R has something to do with the pathogenesis of the membranous nephropathy? What do you think about it?
Yeah, I wish we knew more about the protein itself. I mean, it seems to be a receptor for these small secreted enzymes called phospholipases, and it may play a role in the podocyte of trying to detoxify them, to make sure that these enzymes don't damage the podocyte membrane. But really, there's no evidence that the antibodies disrupt the function of the protein to cause disease. I think it's more likely that all these proteins that are turning out to be the antigens in membranous nephropathy, in the rat model, it's megalin. In this congenital antenatal form, it's neutral endopeptidase. In most of primary membranous nephropathy in adults, it's PLA2R. And these are just all big transmembrane proteins that seem to be sitting in the right place at the bottom of the podocyte. And they have multiple extracellular domains that can serve as the target epitopes for antibodies. So they might just be the right protein sitting in the right place. So whatever starts the process of antibody generation, we don't know why humans develop an autoimmunity to PLA2R, but once you have those circulating antibodies, they enter the kidney, they enter the glomeruli, they percolate across the glomerular basement membrane and attach to their targets. And small immune complexes are probably formed. There must be some mechanism that these antigens and antibodies are shed from the cell into the space, the matrix below the podocyte. And then over time, they grow. And so what is it that damages the podocyte? So immune complexes stimulate the complement system. And complement can help attract inflammatory cells. So when C3a and C5a are generated, they're a signal for circulating neutrophils and macrophages and other immune cells to come by. That doesn't happen in membranous nephropathy because once those fragments of complement are generated, they're basically washed out with all the other proteins into Bowman's space, and the immune cells never see them. But what does happen is the terminal complement components, the C5b through 9, they form a big channel, a pore that inserts into the podocyte, and calcium can rush in, and a lot of biochemical changes happen in the cell that make it difficult for the podocyte to maintain its interdigitating foot processes. So the cell becomes simplified, the slit diaphragms are disrupted, and basically the protein leaks around the podocyte, and that's what causes the proteinuria and all the nephrotic syndrome. So again, we don't think that the function, the biological function of PLA2R is important. We just think it's a big protein sitting down at the bottom of the podocyte that acts as the target and then causes this complement-mediated damage.
What you discovered was a milestone because it changed not only our understanding of the disease, but how we treat these patients. So I'm going to ask you about my next question is regarding the treatment. I mean, using immunosuppressive therapeutic regimens is not like something good for the patient all the time. So deciding which patient should have it and which patient should not is very important. And based on your discovering about PLA2R, seems that many patients have changed the course of the disease. So my next question is in this regard. Are anti-PLA2R titers useful in the prediction of disease outcome? So we should measure this antibody in all our patients?
Well, I think it's certainly a good screening test. If a patient has it, that's pretty specific for membranous nephropathy. But your question is, can we predict outcome? And I'd like to highlight what the field of membranous nephropathy was like before we had these biomarkers. So clinicians who were deciding whether or not their patient needed this powerful, immunosuppressive therapy would have to look at the level of proteinuria, and these patients may have 10, 15 grams of protein, and then ask, is that patient getting better? Is the protein going down from 10 to 5 to 3? Or do we need to give them immunosuppressive medicines? So the problem with the clinical approach is that it takes a very long time for these changes to get better. I mean, you can imagine that these deposits are forming very slowly, and the podocyte gets disrupted, and it takes a long time for the process to stop and the glomerulus to clear these deposits and for everything to get back and the slit diaphragms to form again. So it may take years before the proteinuria goes away. And it's very difficult for the clinician to decide, does this patient need the immunosuppressive therapy? If I already gave some, like, for example, cyclophosphamide, which was one of our first treatments, is it working? It's a 6-month therapy with prednisone, with alkylating agents and steroids. Did it work? Is the protein getting better, or do we need even another treatment? So there was a lot of indecision in treatment of membranous nephropathy because it's such a slow disease to evolve. So now that we have these antibodies, what we and others have shown is that the antibody response actually precedes and predicts the clinical response. And we learned this through collaboration with the Mayo Clinic and Fernando Fervenza, who gave us some of his samples of patients with membranous nephropathy who had been treated with rituximab, the B-cell depleting agent. And he had baseline samples, 3 months, 6 months, et cetera. And basically, we just compared antibody levels, anti-PLA2R levels, and we saw that the antibodies came down in many patients. And we looked at the protein response, and we found that the protein also came down, but after the anti-PLA2R. So there are basically 2 disease courses we could follow: the serologic or immunologic course, measured by the anti-PLA2R antibodies in the circulation, and then the clinical response or proteinuric response. So basically how I would answer your question is, if you have someone who has very high levels of anti-PLA2R, it's probably going to take a long time for those to go away, either spontaneously, which happens in about 30% of patients, or with immunosuppressive treatment. So you can imagine that that patient will have disease for a long time. If the levels are low, then the question is that patient at the start of their disease? If we check the levels later, will it be going up? Or maybe that patient was caught near the end of their disease, and the next level we check will be a little lower, and then maybe we could wait to see what happens clinically. So I think it's a good predictor of what's going to happen with the patient in terms of their proteinuria and their clinical disease.
So you will monitor the patient following the levels, the titers of PLA2R while the course of the disease as well, right? So, and another question is about there are many cases described like spontaneous remission of membranous nephropathy. So another question is in this regard, could PLA2R antibody status be useful to predict spontaneous remission of membranous nephropathy?
So I think that's a good question. There are not a lot of studies out there specifically looking at antibody levels and do those patients go into spontaneous remission. So I think the important thing is to know two things. And pathologists can help us by looking at the kidney biopsy and saying, okay, we've identified the pattern of membranous nephropathy, and maybe it looks like a primary case or an idiopathic case because there are no other subendothelial or mesangial deposits and no tubulo-reticular inclusions. But then the question is, if it's primary, is it associated with PLA2R or the second antigen that's been identified, which is called THSD7A? So many times pathologists will stain the biopsy and find that there's PLA2R within the deposits. So IgG, complement components, and PLA2R are all there. So that tells us that the patient has this type of membranous nephropathy. And then you can ask, are there antibodies in the circulation? And if there are, then the patient has both probably clinically active disease and immunologically active disease. But there are cases in which the pathologist will tell us that there's PLA2R in the kidney, in the deposits. It's a normal protein, but the pathologists say that this antigen is aggregated into these deposits. And we'll check the serum, and the patient doesn't have anti-PLA2R antibodies. So that can happen in two different circumstances. Potentially, that could happen very early in the disease. When the immune system's making very low levels of the antibodies and everything is being trapped in the kidney, there's so much antigen that the IgG, the anti-PLA2R, is all being finding its target, and there's not much left in the circulation. So in that case, we'd expect that the patient really doesn't have severe disease. They probably don't have much proteinuria. Maybe the deposits will be stage 1, the smallest type of deposits in the kidney. In my experience, that doesn't happen very much because just like it takes a long time for the disease to go away, we think that it takes a long time for this disease to develop. And we know this from kidney transplant patients who get recurrent membranous nephropathy. That their clinical disease starts a year after transplantation, even if they're known to have circulating antibodies. So by the time a patient comes to us as nephrologists, they've probably had disease for 6 to 12 months, and they're likely to have the antibodies in the circulation if they were there. So what is the other case in which you might find the antigen present in the biopsy but not have the circulating antibodies? So that would be at the other end of the disease, where at one point there were circulating anti-PLA2R, but they've come down and disappeared, and the patient is still proteinuric, they still have clinical disease, but we would expect that to get better. So you can think about, again, two courses of disease, the immunologic or serologic, which has the antibody levels go up, they plateau, and then they come down with time. And then the clinical disease, which follows that. So again, the proteinuria will go up, plateau, and then over time will come down to a partial or complete remission. So in many cases, when we find a patient who has membranous nephropathy on biopsy, whose immune deposits have PLA2R, and they don't have antibody in the circulation, those are the patients that are likely to spontaneously remit. There haven't been any big studies, as I said, but in my practice, that's what we see.
Yeah. Great. So not finding the PLA2R in the circulation, it could also be a false positive. What do you think, depending on how you measure the titers, or are there different ways to do it?
Yeah, so worldwide, there are several assays, but one company markets an ELISA, and basically you can get a level that's anywhere from 2 to 1,500, and technically, if it's under 14, that's regarded as negative, but I think a number of investigators have shown that you really have to have levels that are undetectable, less than 2, to really call that negative. So that's the ELISA test. There's another test that uses transfected cells on a little slide, and you add the patient's serum and you look, are there anti-PLA2R antibodies that are binding to the cells, similar maybe to the older ANA assays, and you're asking more subjectively, is there a titer of 1 to 10, 1 to 100, 1 to 1,000? So those are the two main assays out there. But you're right, there are cases where we're a little unsure about, is this a truly negative test? Is it just a very low level in the circulation that might be missed in certain circumstances? So it may be worth repeating that to see if the levels change over time.
Thank you. And okay, you already talked about the measurement before and after 6 months. And what about thrombosponding? Because you mentioned about the 70% of cases may have PLA2R, and we still have 30% that may present other things. So thrombosponding came also like a very exciting, promissory protein. So could you tell us about thrombosponding?
Yeah, so we and others had noted that there were certainly cases of membranous nephropathy that interacted with other proteins. So when we did our Western blot using patient serum against the glomerular proteins, occasionally we got some slightly different bands. And we actually first noticed in 2010 that there was a patient with prostate cancer who had membranous nephropathy, and his serum identified a band of a similar size that wasn't PLA2R. And we actually published this in a review article I wrote in Seminars in Nephrology on the relationship between membranous nephropathy and malignancy. So if you dig up that article, you can see the first evidence we ever had of THSD7A. But over time, we found a few more cases, and it turns out that the Gérard Lambeau's lab in France, and he was working with a student from Rolf Stahl's lab in Hamburg, Germany, and the two of them, Nicolas Thomas and Gérard Lambeau, also were looking at this other antigen. And it turns out that in maybe 2013 or so, we were on a phone call together, and we realized that we'd come up with the same protein as target antigen called, and again, it was one of these proteins on the lists of mass spec that we got back from the University of Louisville, and it was called the thrombospondin type 1 domain containing 7A. And both of us had found this protein, so we decided to write the paper together. So that was the second description of an antigen in membranous nephropathy, the THSD7A. And again, this was a protein that really there wasn't a lot known about. I think there were maybe 4 articles in the literature, nothing related to the kidney. So we had to learn a little bit about this protein, and it's also a large transmembrane glycoprotein that has a lot of domains that stick out from the bottom of the podocyte that serve as targets for these autoantibodies to THSD7A. It may turn out that this is a more interesting protein to the podocyte. There was a recent paper from Hamburg, Germany, a few months ago in JASN, Journal of the American Society of Nephrology, that shows, and the senior author here was Catherine Meyer-Schwessinger, and basically she and her group showed that this protein THSD7A sits immediately beneath or basal to nephrin in the slit diaphragms. So what is it doing there? We're not sure. Is it directing the formation of the slit diaphragms? Is it working as an adhesion molecule? There's also data from the Hamburg, Germany lab that if you inject mice with either human antibodies to THSD7A or rabbit-derived antibodies, you can quickly cause proteinuria. And they don't necessarily find complement. So it may actually be that these antibodies are somehow disrupting the podocyte and the slit diaphragm to cause proteinuria. So I think this is a more intriguing form of membranous nephropathy because these autoantibodies may actually be disrupting the biological function of this molecule.
Yeah, we have seen many, not many, but we don't have that many cases with the mark with the positive with thrombospondin, but several that we have seen have cancer associated with cancer. So it's very exciting to find out the relationship between cancer and thrombospondin. Mm-hmm. And also, some of them are also they receive some kind of chemotherapy. So, and the membranes look more like drug-induced membranes, and they are positive for thrombospondin. So it's very exciting to find out if this is drug-induced membranes, positive for thrombospondin, or cancer-associated membranes. So very, very interesting. So what is the future of the research in membranous nephropathy, and what do you expect, or what are you doing now? Can you tell us?
I think we're still, I mean, we'd also love to know, you mentioned 70 to 80% are PLA2R associated, maybe 3 to 5% are THSD7A thrombospondin related. We and other people are looking for what are those last 15 to 20% of patients. We certainly find some bands on Western blot that we're trying to figure out what those are. There's a recent publication actually this month from the Mayo Clinic, and Sanjeev Sethi, the pathologist, and working with Pierre Ronco, have identified a protein called exostosin as the biomarker, perhaps target antigen in class 5 lupus nephritis, membranous lupus. So we hope to use similar techniques to maybe figure out what all the other antigen targets are. But I think, biomarkers are great, but we'd really like to understand where, how to treat these patients more specifically, and what the mechanisms are in the human. There's no good mouse model of PLA2R associated disease, and we can't necessarily rely on the rat model that uses megalin as the target to tell us everything we want to know about treating human disease. So one of the projects, and this is how I got to know you, Carmen, is through the Nephrotic Syndrome Study Network, or Neptune, which takes human biopsy samples and microdissects out the glomeruli in different diseases. So we have all the gene expression data. And one of the things we've done recently, working with Toronto and Michigan and others, is to come up with a gene signature set. So those genes that are upregulated in membranous nephropathy that aren't upregulated in other diseases like minimal change disease or FSGS. And interestingly, they turn out to be a lot of podocyte genes, some of which I'd never heard of as podocyte genes. So we're trying to fit the pathways together to figure out in the human what pathways of disease are active, what pathways of repair of the cell and the glomerular-based membrane are active, in order to better come up with ways to treat this disease. So those are some areas that we're still interested in, but the future is certainly bright and there's lots of areas to study.
Thank you so much. And I'm also very excited to participate in this Neptune project, and I wish the morphology could give us more, but I think it's time to recognize the limitation that morphology can give us and put it together with all the findings you have mentioned, genetically, biochemical, and probably give a little more information that may help the clinicians how to treat these patients in the future. And so I think these are all the questions I have for you today. And thank you very much for, and I enjoyed a lot talking to you about this science, and Thank you. So do you have anything to say for our audience in Latin America?
Well, thank you all for your hard work, clinically and scientifically, and I hope that everyone remains curious as to how these diseases occur and how we can better diagnose them and treat them. And we certainly need all the minds, the curious and the creative minds, to help us solve these diseases, these exciting but troubling glomerular diseases, because the patients need our help. So thank you for watching this, and best of wishes to all of you.
Thank you.
Thank you, Carmen.